Good morning to all. I am Francesco Guarnera, a researcher involved in FAIR project. Today, I will introduce briefly some of my works related to the studies of brain image analysis with the use of artificial intelligence. Medical image analysis is often affected by human bias, especially in manual or single expert diagnosis. Subjective interpretations can lead to inconsistent classification of pathologies. This variability introduces uncertainty, reducing the reliability and reproducibility of medical finds. Artificial intelligence-driven analysis, when properly designed and validated, can mitigate these biases and promote objective, fair, and reproducible outcomes. Moreover, generative AI techniques enable temporal projection, supporting longitudinal monitoring of neurodegenerative disease and the integration of multimodal imaging data for a more comprehensive understanding of brain pathology. Some tasks related to the brain image analysis are segmentation, that has the goal to delineate the brain structure and lesions and then to quantify damage. Brain progression, that have the goal to model temporal evolution of the brain, useful to the early detection of neurodegenerative disease. Brain aging, to quantify the difference between chronological age and biological age brain, useful to quantify neurodegenerative disease status. And atrophy map, to calculate the loss of matter correlating the loss to the disease of cognitive anomalies. Now, I will present some works related to the task described before. One of them is related to the automatic segmentation of brain lesions in patients affected of multiple sclerosis in MRI. Images in the slide show the task difficulties with two specialist raters producing totally different segmentation masks. In this first work, we presented a pipeline for a longitudinal study based on a proper processing and based on a UNet enhanced with the addition of attention mechanism for a longitudinal studies that represent an important boost with respect to the state of the art. In the last 2 years, other 3 works have been published in the field. The first 2 present the same architecture described in the slide and based on a diffusion model, working on a one-step segmentation of lesion without any patient's knowledge. With the second one that had a partition strategy before, with the generation of the mask for each partition, and a merging strategy at the end of the architecture. The last one on this field, the field of multiple sclerosis, is a 3D network recently published based on a hierarchical multiscale learning, deep supervision, and arthrospatial pyramid pooling. Also to mention the dataset that we built during the last 3 years and that actually represent the state-of-the-art benchmark in MRI lesion segmentation for multiple sclerosis, and the challenge that we organized, me with my group, in 2024 at the International Conference on Pattern Recognition. Another important task faced was the use of generative AI to produce the progression of brain of MRI. This task permits to project in the future the structure of brain and to find some development of structural change of the same. We conduct studies on a dataset composed of Alzheimer's disease and healthy patients. Our architecture is based on a diffusion model conditioned on the baseline image at time point 1, patient metadata such as cognitive status, and the difference in months between baseline and the follow-up image that we want to generate. The image on the right shows the generated images by our pipeline and the difference with the real follow-up. Recently, we submitted also the 3D version of the same architecture, which has the challenging goal to produce the full volume of the future MRI. The task of brain aging is strictly related to the calculation of predicted age difference, or PAD. PAD is defined as the gap between brain age and the chronological age, often used to investigate neurodegenerative conditions. In this field, we proposed the synthetic brain age, a deep learning model designed for predicting brain age. It employs a domain randomization techniques based on the famous synthetic generative pipeline and following by a 3D densenet able to produce the age prediction. Finally, we faced the task of atrophy mapping calculation, crucial for evaluating brain disease and analyzing neurodegeneration. In this study, we propose a simple 3D UNet able to calculate the map in an average time of only 3 minutes with respect to the 17 minutes employed from the actual baseline used for the task. Finally, thanks to FAIR for the opportunity to work in this important field, and thanks to all for the attention.